Okay, our next speaker is Jouni. Uitto. You saw him yesterday, so he doesn't need much introduction. Jouni. Let's see, are you muted? Here you are. We're still not hearing your voice, which is odd since you usually are not shy about speaking.
Certainly not. And thank you, Sharon. While I get this this presentation online. Here we go. Are we okay now?
Very good, yes.
Okay, thank you, Sharon. So I'd like to give a brief presentation, actually, which extends very nicely what Viola was just talking about. In other words, I want to tell you what kind of work goes to preclinical drug development for PXE. In other words, what do we have to do before it can be tested in humans? And I'm using as an example one kind of recent compound called INS 3001. It's a kind of mysterious name, but all these drug companies have this kind of code for their products. So basically, there are a number of preclinical activities which are leading to clinical trials. And in fact, there are some early clinical trials already in place for treatment of PXE. We heard about the etidronate. There is the inorganic pyrophosphate concept going. And many of these trials actually have focused on the attempts to increase the pyrophosphate levels, either by oral administration to the patients or by using so-called, on the right-hand side, TNAP inhibitors, which TNAP breaks down pyrophosphate. So if you inhibit TNAP, you get presumably increased levels of PPI or inorganic pyrophosphate, and you will hear from Dr. Hubert Cho about that approach a little later. But there are also concepts which have nothing to do with PPI. Basically, the concept is that you somehow prevent the deposition of the mineralization. And mineralization, basically, it's hydroxyapatite crystal deposition. And if you are able to prevent that deposition, it doesn't matter what your PPI levels are. And that's what this drug, INS 3001, is representing. It's actually developed by a Swiss company, Inositec, it's based on the notion that there is a naturally occurring substance known as phytic acid. It's present in nuts and seeds, and it's known to have some anti-mineralization properties, but relatively weak so, and it is relatively quickly cleared from the blood. So this company has modified this naturally occurring molecule by a number of chemical changes, which then hopefully increase the half-life and the efficacy of this potential drug. And here are sort of the structures of the phytic acid on the left and the modified INS 3001 on the right. So they look very similar, but have some differences. Now, what do you first have to do? So first of all, you have to have some idea that this drug may be useful. You don't just pull any drug from the shelf, but you have some kind of suspicion that this drug might prevent calcification by some mechanism. And this INS 3001 is based on the notion that there are small protein particles in the serum, in circulation. And these small particles then precipitate to form calcium crystals. So in other words, the calcium deposition. Now, this naturally occurring molecule, which is here listed at IP6, in the middle panel is known to inhibit that precipitation. But now the question is this INS 3001 more efficient? And actually, if you look these curves here, so at a 10 micromolar concentration in the middle, this naturally occurring compound, it takes about something of the order of 4 hours the precipitation of calcium to take place. But now the red line shows that this new improved molecule, it takes actually about 3 to 4 times longer to calcium to precipitate. So you have a slower precipitation, potentially inhibiting the mineralization. Now, next, and so that was sort of a test tube experiment, but then next you see tissue or cell cultures. And in this study, the company used smooth muscle cells, which are cultured from arterial blood vessels. And you culture them, and there's a way to mineralize those cells by inducing the mineralization by calcium phosphate. So on the left, you have the calcium phosphate, that's the gray panel. And then you start giving those cells, you incubate them in the presence of this INS 3001 and see if you can inhibit the mineralization. And those red panels there, or red graphs, they show that when you go to as little as 1 micromolar concentration of the drug, in cell culture, you completely inhibit the mineralization. So it looks so good in cells. So basically, in fact, if you look at the half-life of this new molecule, it's about 78 minutes versus the 8 minutes, the naturally occurring one. So it stays much longer in the circulation than the naturally occurring anti-mineralization compound. There are also studies which have tested this INS 3001 in a mineralization or calcification model, very different from PXE. Basically, you give high concentrations of vitamin D3 and a molecule known as adenine, and you get calcification of the blood vessels. And in this model, actually this INS 3001 is able to inhibit the calcification of the blood vessels. So we know now that the calcification in vitro, either in test tube or in cell culture, or in vivo in a mouse model of a calcification, they showed that this INS 3001 is potentially helpful, but its efficacy in tissue calcification, PXE, has not been demonstrated. And that's the little study that I'm going to present shortly. So basically, we used those APC6 mouse models of PXE, which actually Viola Wiebe developed first, our independent APC6 mice. And we use a mineralization of the skin in a structure that surrounds Vibrissae. There is a dermal sheath of Vibrissae, and that becomes mineralizing at about 5 to 6 weeks of age, and reliable mineralization is by 12 weeks. So here are the mice, and instead of like Viola was shown that you kill the mice and look for mineralization, we can take a little biopsy from the whisker area of these mice, and then use the histopathologic analysis, as Viola was showing, for mineralization, or we can measure by chemical assay the calcium content of the muscle skin. So this is something that you can follow in the mice in a progressive way. And actually, here is an example. The mineralization is progressive. On the left is normal mice at 6 months of age, and there is no mineralization in this muscle skin surrounding the Vibrissae. But if you look on the towards right, there are 6 weeks or 1.5 month age, you start getting mineralization, those dark precipitates on the periphery, and 6 months you get much more, and 22 months, you have extensive mineralization. So you can quantitate this mineralization either by scanning the color, or you can actually solubilize those little biopsies and measure the amount of calcium, exactly like Viola was showing. So in our study, we treated these ABC6 knockout mice. So basically they are PXE mice, which recapitulate the features of human PXE. And we administered this 3001 two different ways, either by osmotic pump, so you have a continuous delivery, or by injecting either 3 times a week or single week injections. And then we look into the histology and measure the calcium content. We also can measure the plasma drug concentrations in those mice. Now, here is an untreated mouse, which served as a control. It's a PXE mouse, so you get the mineralization. And now if we start using the pump with 0.16 milligrams per kilogram per day, there's much less mineralization. If we increase it 5-fold, there's very little mineralization. And in the 4 milligrams, there is no mineralization at all. We can also show that by injections at different doses and different concentrations or amounts of this drug, we get inhibition or mineralization. And we can also quantitate that by calcium content measurements, which here shows in that area, in red outlined area, that increasing the amount of drug, you get less mineralization, and with as little as 4 milligrams per kilogram in the middle, you could go as low as in the wild type mice, in normal mice, the level of calcium on the left. And also we can show that the injections, of course, give you a single bolus, very high levels of the drug in the plasma, but continuous pump actually gives you very significant levels, which should be enough on the long term for treatment. So basically, this shows that this drug, INS 3001, prevents skin manifestation of in the PXE mice, prevents the calcification, and it probably could be a good treatment for PXE. However, there have to be lots of work being done. First on the animal models, look into the toxicity, long-term effects, any side effects, that sort of things, before it can be applied to early human clinical trials. So it's a long journey for any of these drugs to be developed and take several years to have something which we can with confidence apply to humans. This work was actually done in the PXE Center here at Jefferson in the laboratory of Dr. Kwali Lee, who's there at the bottom on the left, and the other individuals participating are shown here. So I'll be happy to answer any question, but this was supposed to illustrate how much work really goes to developing a safe and efficacious drug for a condition like PXE, which is currently intractable. Thank you very much.
Great. Thanks very much, Jouni. And so questions. Dina asks Jouni, how many people are taking D3 right now, and any concern at this point?
I don't think so if it's within the kind of recommended levels. That mouse model that I was talking about, which gives you early calcification, that's a very high vitamin D levels, but for the amounts of vitamin D, I do not see any problems.
So Jouni, I'll ask for you to comment on, while we're hearing lots and lots of potential treatments today, and you know well that we heard years and years ago that maybe it was vitamin K, and then maybe it was magnesium, and maybe it's this, and maybe it's that, what's your take on what people should be doing, taking right now?
As I tried to indicate, it takes long time to get any of these through the preclinical development steps to get to the clinical trial stage. But there, as you know, there have been a couple of clinical trials already. The magnesium trial was recently published, and the pyrophosphate trial is early stages ongoing. The Wilco was talking of the atidronate trial, which obviously needs some follow-up before it can be recommended for routine use for PXE patients for safety and efficacy. But so at this point, I think just normal diet, healthy diet. The magnesium trial, unfortunately, was unequivocal in the result that we could not quite make sense whether it works or not, in part because the group of patients was relatively small, and there is not really good way of measuring in humans without going very long term whether the drug is useful or not. Magnesium, many people do take magnesium. There are FDA recommendations, 425 milligrams per 75 kilogram person per day. Normal diet usually gives about that range.
And then the other thing, and I think maybe Hubert might make this point as well, is that if people decide to do their own private clinical trial, then they probably aren't eligible for some trials. So for example, a couple of these have already said that if you're taking excess magnesium, then you can't participate in the upcoming trial. So
yeah. Yeah, and that also complicates all these clinical trials that are ongoing, because obviously while PXE, it's not very rare, it is a rare, and one person cannot obviously be in two different clinical trials at the same time. So we have to carefully prioritize which kind of trials we should participate and which would be the most kind of reasonable approach to develop these drugs.
Great. And we have a question from Susan Ritchie. Susan, you can speak.
Susan, are you there?
Yeah, she's muted. I think she's working on getting unmuted.
There we go. Thank you very much. I have been reading and hearing a lot about vitamin K2 supplements. Can you address that?
Yeah, so there was, for a while, there was a notion that maybe vitamin K is somehow deficient, or the PXE can be counteracted by vitamin K. This notion was actually based on observations in a very few patients who have bleeding tendency at the same time as they have PXE. And turns out that it's a completely different gene. It's not the ABCC6 mutations, which account for about 95% of all PXE patients. There are very few patients which have PXE-like clinical presentations in their skin, who also have bleeding tendency because of the gene defect that participates in the coagulation pathway. So it's not something that is going to work in most people with the PXE at all.
Okay, thank you.
All right, any other questions? Okay, well, thank you very much, Jouni, for taking part of your Sunday with us.
Thank you.